Hi, I'm Sam.
I'm Apeek.
And we're UTAs for Dr. Ashmore. And so this is the third step of glycolysis. It's the rate determining step. This is the enzyme phosphofructokinase, abbreviated as PFK. And phosphofructokinase has two sites on it. One is the active site and one is the regulatory site. So in both these sites, ATP binds to each of these sites. The active site has a higher affinity for ATP, so if you have a smaller amount of ATP, it'll first bind to the active site and it'll react here. But if you have a larger amount of ATP, it'll start to bind to the regulatory site and it'll slow down the overall reaction. So in this way, ATP acts as a substrate as when it binds to the active site, but also as an allosteric regulator when it binds to the regulatory site. So in our PFK enzyme, we have the active site and the regulatory site. So imagine if you have a lower amount of ATP, all the ATP would bind to the active site first. The active site has a higher affinity for ATP, or the ATP has a higher affinity for the active site. So all of the ATP available will bind to the active site until the active site is full. The moment the active site becomes full, ATP will start binding to the regulatory site, and once ATP starts binding to the regulatory site, the overall process of the reaction definitely slows down and not as much, it slows down the overall reaction. But this will only happen if the active site is totally full, because the first place that ATP will bind to will be the active site, as it has a higher affinity for the ATP. The regulatory site is used for excess of ATP, so in that way ATP is used as an allosteric inhibitor or an allosteric regulator as well as a substrate. So the reason the active site has to have a higher affinity for the ATP than the regulatory site is due to the fact that if it didn't, in a situation where the regulatory site actually had a higher affinity, all the available ATP would go to the regulatory site, and once the regulatory site was full, then it would start filling the active site. But no matter what, in that situation, the reaction will be slowed to a level where it's not practical for making energy. So in a world where the regulatory site has a higher affinity for ATP, that basically means the reaction is not occurring at all, under any circumstances, no matter how much ATP that you have in the system. So the next step in the process is the citric acid cycle. The main products of the citric acid cycle are NADH, FADH2, and 2-ATP. The most important products are the NADH and the FADH2. These are both electron carriers, and they are used later on in the electron transport chain.
Yeah, these electron carriers shuttle electrons to the electron transport chain, which uses them to create a proton gradient, which is then used by ATP synthase to make ATP. And if there's too much ATP,
so when there's an excess of ATP, the ATP will go back and inhibit the citric acid cycle at a certain point, kind of like feedback inhibition.